Hello, Riversiders. Today's lesson is our last lesson, last section on life science, which is DNA, the structure behind all of that heredity that we learned in the last unit. So, as usual, we're going to jump right into sharing the screen. DNA stands for deoxyribonucleic acid. Long word, just means, well, it just describes what it is. It's got nucleic acids and a backbone of riboses that are missing an oxygen, a sugar backbone. So the shape of it is, as I just said, a backbone of sugars. There we go. A backbone of sugars. That's these little 5-carbon sugars or riboses. Glucose is 6-carbon, but whatever. A backbone of these sugars and with some nucleic acids coming off the side. These riboses are missing an oxygen, so it's not stable as a single strand of just riboses with nucleic acids. It needs a second, the nucleic acid needs to bond to something, and so it becomes a pair. You have one strand going this way, one strand going that way, and they connect to be a nice stable structure. And then once connected, it'll spiral up in this double helix shape that that you see right here, the classic image that we know of for DNA. You'll note that there are 4 different nucleic acids. There's thymine, cytosine, adenine, and guanine. You don't need to know them. What you do need to know is that A's, adenines, and T's, thymines, always bind with each other. And cytosines, C's, and guanines, G's, always bind with each other. So C's and G's bind and A's and T's bind. Whatever mnemonic you want to remember that, go for it. But it's a very important part of the way DNA works, because in that binding, it gives us the ability to both replicate itself. It's self-replicating. It can make copies of itself. How many substances can do that? But it can. And it also gives it the ability to be a template or a code that codes for proteins. I'll hopefully explain all of that. We're going to learn about DNA replication, DNA transcription, and DNA translation. And that's what this unit's about. So A's, T's, C's, and G's. So since they always bind in the same pattern, if this section were unzipped, A, T, T, A, T, T, C, G, T, A, G, G, blah, 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 you know what would bind with it, because the A would bind with a T, and the T would bind with an A, and the C would bind with a G, and the G would bind with a C. I don't want to spend too long on that because we'll run out of time. So anyways, transcription. So that's how it replicates, by the way. It unzips. Let me jump down to replication. Here we go. It unzips. Zip. Some protein comes by and unzips it. And now you have these 2 single strands of DNA, which are not stable. So complementary nucleic acids will bind. A's with T's, C's with G's. And then at the end of it, you end up with 2 strands that are identical to each other. This is where mutations come in, which we'll talk about later this unit. If you have enough energy, you can ram the wrong pieces together, an A with a G, say, and that's not good. So transcription is the process by which we transcribe DNA in the nucleus into RNA, ribonucleic acid. Notice it's not deoxy, so it's stable in a single strand form. So we're gonna transcribe it from DNA into something that's stable enough and small enough to leave the nucleus. So we transcribe it into RNA. And this is more or less how it happens. It unzips just a section, not the whole strand of DNA. Just a section unzips. And as it unzips, this little protein goes down and helps bind the complementary base or nucleic acids to it. The only difference is between DNA and RNA is RNA is stable as a single strand, and it doesn't have T's, it has U's instead. Wherever a T would be, it'll be a U. Not a big deal. You'll learn this in genetics if you study genetics in high school or college. So here we go, we're transcribing it. And now that little section of DNA is not our DNA, it's RNA, single strand. So it can go out of the nucleus, and it does. It leaves the nucleus and gets carried by the endoplasmic reticulum. Remember, way back to the cell unit. It's carried by the ER, the endoplasmic reticulum, out to where the ribosomes live. So it's transcribing, and then the ribosomes translate the DNA, or in this case, now it's RNA, into proteins. That's a relatively complex process. I'm going to do my best to do a summary here for you, but I'll also explain it in class. So here we are transcribing a section of RNA. And then, so that's transcription, lots of images of it. And then it'll get translated out here. Now you get the section of RNA, it's a single strand. And every 3 nucleic acids is a code word for a different protein, a different amino acid. A protein is just a string of amino acids. So we've cracked the code, and it's the same code for all life, which is really wild and cool. But we've cracked the code. So these 3 are code for this amino acid, and then those 3 code for that amino acid, and it'll make it'll string them along here, and string together a bunch of amino acids, which then get morphed and shaped into a protein that does something. Here's the code. You do not need to know this. It's just cool, so I put it on here anyways. So like TTT is the code for phenylalanine. You don't even you probably can't even read a lot of these abbreviations. TCT is serine, or proline down here. But you can start to see that if you were to change even just a single nucleic acid, a single one of these A's, T's, C's, or G's, you can vastly change what the thing is. If you change it just by just substituting one for another, then you might change which of these it is, which sometimes is bad. Sometimes, like over here, it's not going to change anything. It's all going to be arginine. But up here, you might stop the process entirely. Over here, you might get a lysine where you had a different one. Like you might change what you get. And that's the basis for evolution. Some mutations, some changes in the A's, T's, C's, and G's, and now you have a slightly different shaped protein, or maybe the protein doesn't work at all. And that slightly different one could be more effective, it could be less effective. It depends on the environment. So I know I'm going very quickly. Mutations, it's just a sudden change in the structure of the DNA. It's ultimately the necessary for all evolution. All differences in creatures, between one creature and another, and between all humans and all creatures within a species, is a result of evolution, or is a result of mutations. Evolution couldn't happen without mutations. And most mutations have little or no effect on the organism. Sometimes it's in a section of DNA that you don't even use. Some mutations are harmful and cause the cell's death. Some mutations might be really harmful and cause the cell to like grow uncontrollably. That we call that cancer. Anyways, something that causes a mutation is a mutagen, generating a mutation. The first one to show it in all of its genes is called a mutant. Anyways, the worst place to get a mutation would be in your gametes, in your sperm or eggs, because If you get a mutation anywhere else, it's just going to affect that one cell. So what? That one cell's, say, green. You won't even notice one green cell amongst the trillions and trillions of cells you have. But if you mutate an egg or a sperm to be green, that's going to give rise to every cell in the baby. And so the baby will be a green baby. So there are different kinds of mutations. I'm probably not going to stress this terribly, but there are point mutations which just affect a single nucleic acid. Substitutions being the least harmful because you just change what it is a little bit. Insertions or deletions could be really bad because now you're going to change everyone after it. Like, so if you have one sequence, if you substitute, all the rest stay the same. But if you add one in, now it changes the every 3 bases. So everything downstream of that one mutation is going to be different. So those are really drastic ones, either insertions or deletions. You could also argue chromosomal mutations are pretty drastic. That's where whole chromosomes get messed up. You have insertions and deletions there. You have substitutions there. You have inversions and translocations. But the biggest one that you probably should know about is called non-disjuncture. That's when normally during meiosis, we pull the DNA, the 2 different chromosomes apart. If they don't pull apart, then one sperm or egg is going to have an extra chromosome and one is going to have too few. And this has a really major impact on a lot of the way the creatures lives. Down syndrome would be an example of that. So would Turner's or Klinefelter's. We are particular in the balance, so we need just the 23 and 23, so the 46 chromosomes that we have. Too many or too few, and we don't do so well. So that's the better part of this unit. I know it was very quick. I need to keep these quick. Let me know if you have any questions. I will happily do a deep dive in any part of this unit, and teach you more. Until the next one. See you later, guys.